All right, so just quick review of what we've already been mentioning is you've got your respiratory system doing these basic things. There was pulmonary ventilation, which was basically bringing the air down into the lungs where you can actually let the oxygen diffuse into your bloodstream. Then we had all those conducting zones, all the little paths going down the trachea, the bronchi, terminal bronchiole, finally into the alveoli. And the alveoli is where we had the external respiration, where we actually had the movement, the diffusion of oxygen from the alveoli into the bloodstream. And we saw that that was driven by the difference in partial pressures. The partial pressure of oxygen in the alveoli was around 100 millimeters of mercury. The partial pressure of the oxygen in the blood that's returning from the tissues to pick up more oxygen is only around 40 millimeters of mercury. So we had the oxygen move from the alveoli into the bloodstream. And then the blood is going to go off and transport it. And we're going to be looking at the other side, what happens when the blood arrives in the tissues in a few moments. We also kind of talked about just the basic anatomy, ways that you kind of deal with dust that you're breathing in. She probably would not be happy, but I'm going to draw big nose hairs on here. You know, there's nose hairs, the vibrissae, that are kind of just blocking out big dust as you're breathing it in. Then as you're going into the respiratory passages, as well as the lower respiratory tract, those are all covered with that epithelium that has mucus and little cilia that are sweeping along. So most dust gets stuck in that mucus and then swept either to the back of the throat if it's in the upper part, or up the mucus elevator or escalator to the back of the throat. Either way, once it gets there, you swallow it and it's kind of out. We also talked about even once you get down into the alveoli, there are going to be alveolar macrophages, also known affectionately as dust cells. And those are on patrol and they can eat any little stuff that ends up down in there. And you're breathing in all sorts of stuff. Even if you don't think about it, there is so much particulate matter in the air around us. Every car that's driving down, or that used to drive down the street before all this sheltering in place, spews out tons of little particles. So even you don't have to be a smoker or live in some really polluted place to have lots of crap coming down into your lungs. So you constantly have these processes going and cleaning out the stuff in there. What other things kind of review? We talked about the cartilage, which helps keep your airways patent, keeps them open so they don't collapse in. You have those cartilage, so C-shaped cartilage rings down your trachea, cartilage rings down your bronchi, until you get down to the very small. The little bronchioles no longer have cartilage. Those just had smooth muscle. And we talked about that is under control of the sympathetic nervous system. You know, if you're breathing in something really noxious, your parasympathetic can actually contract those so you don't actually suck it all the way down into your delicate lung tissue. When you want to dilate them, because you're breathing like part of your sympathetic fight or flight, you dilate those so you get less resistance to flow. Because again, the flow of air through these little tubes is defined by the same equations that we saw for the flow of blood through blood vessels. You know, it's going to be pressure. I mean, I'll say delta pressure. The pressure difference on one side versus the other side is going to be, you know, the flow times the resistance. That's the basic equation. For blood flow, it was blood pressure was cardiac output times resistance. For air, it's the air pressure is the flow times the resistance to flow. And again, if we write it the other way, flow, yes, I should probably write it on something different, but it's going to be the pressure divided by the resistance. So this relationship, whether we're talking about blood flow or airflow, it's really useful just to remember that kind of relationship. You increase the pressure difference, you increase the flow. But if you increase the resistance by usually narrowing the tube, you decrease the flow if the pressure doesn't change. So.
Could you talk again about what's happening with asthma?
Yeah, asthma. In asthma, you have these bronchioles constricting. And when those bronchioles constrict, you're increasing, so it's like an allergic reaction, and part of the reaction is to constrict those little vessels, those little bronchioles, those little tubes leading to the alveoli. So the resistance is going up. So now the same pressure differences that you would use to drive the flow of air in and out of your lungs are resulting in less flow.
Okay.
So you're going to have to increase the pressure differences, and it makes it, again, harder to get air in as well as harder to get air out.
Mm-hmm.
So then we talked about pulmonary ventilation itself. Again, the basic idea is you've got the lungs and they're in this pleura, and the pleura has a suction. So when your thoracic cavity gets bigger, the lungs get bigger, and when the lungs get bigger, the pressure goes down. And then from this thing, if the pressure is going down, then now all of a sudden the flow is going to be, or actually I should say, you end up having low pressure here, high pressure out here, so the flow goes in. When you're going to breathe out, it's the opposite. You relax. Again, this is going to come into this COVID thing. Part of your breathing out is that lung compliance, that basic stretchiness of the lungs, and plus the surface tension. The lungs naturally want to collapse. So when you relax your muscles and your thoracic cavity gets smaller, then the lungs are going to get smaller, increasing the pressure inside. So when the pressure is bigger inside compared to outside, the air is going to flow out. So this is a really freaking messy picture here, but again, of all the muscles, which is the most important for driving pulmonary ventilation?
The diaphragm.
Yeah, totally, the diaphragm muscle. So the diaphragm muscle is the majority of it, but all these other muscles in this picture can play a role. So when you're breathing, these also can contract to increase or decrease your chest cavity. When you're doing more of these forced inspirations and expirations, which we're going to see in our lab, these are going to play much more of a role. For quiet breathing we call tidal breathing. Tidal, like, the tides running in and out. The diaphragm is only going up like a centimeter or so, and the pressure differences are only a few millimeters of mercury to drive the normal breathing. When you're doing like a real forced inspiration, your diaphragm might plunge to like 10 centimeters, and you might have a pressure difference of like 100 millimeters of mercury inside and outside to really get the air rushing in. But during quiet breathing, it's just small physical distances and small pressure differences. And and again, inspiration is always active. You have to contract muscles to increase the volume of your chest, therefore your lungs to inspire. But expiration is typically passive. If you're just doing quiet breathing, you just relax and the chest gets smaller and the lungs then get smaller and pressure builds up and air moves out. This is going to come up when we talk about COVID in a moment. Again, compliance. Compliance is that stretchiness and elastic recoil of your lungs. Right? If compliance is compromised, like for instance, in emphysema, your lungs get less stretchy, or in some of these really bad inflammations and pneumonias, your lungs get less stretchy, which means that exhaling is going to be trickier. Getting all the carbon dioxide out is going to be trickier. As long as this compliance is good, when you just kind of relax, the lungs deflate and you clear out the stale air and you don't have a buildup of CO2, which we saw last time means you're not going to feel like, oh, I got to breathe. But it doesn't necessarily mean that you've got enough oxygen. So we'll get back to that in a second. Alveolar surface tension, again, we talked about the water surface tension is pulling the alveoli smaller, and you need to break that with that surfactant, or else you're going to have a lot of trouble increasing the inflating your lungs. So, and pulmonary surfactant. This is the stuff that you are not making when you've got COVID-19. And also preemies. Preemies don't have enough of it, which is why you have to put them on a ventilator. They can't breathe on their own because it's too much work to inflate their lungs without the surfactant. So if a baby is born prematurely, they actually spray its respiratory passages with surfactant and then put it on a little ventilator. So again, force is keeping alveoli open. This is just transmural. This is basically the suction across the pleura because of your chest muscles opening up your thoracic cavity. Pulmonary surfactant breaking down the surface tension. Force is promoting alveolar collapse, that natural elasticity of the lung tissue, as well as the alveolar surface tension that is residual, is making the lungs want to get smaller. Then this is kind of a review of kind of where we left things. We talked about pulmonary circulation here. As the deoxygenated blood goes into the lungs, it's got a partial pressure of oxygen of around 40 millimeters of mercury. In the lungs, where the lungs have 100 millimeters of mercury in the alveoli, there's a diffusion of oxygen from the lungs into the blood. So when the blood is leaving the lungs and going back to the heart to go to the systemic circulation, the blood is now loaded up at about 100 millimeters of mercury partial pressure of oxygen. This is going to be important when we start talking about this part, this internal respiration. This is the internal respiration that we haven't really talked about yet. Oh, I wish I had a stylus from my other computer here. Internal respiration is this stuff happening in the tissue. This part up here we called external respiration. So what I'm going to do is I'm going to take this off now for a second. Stop sharing. So any questions just about the basic review? All right, so now I read this article. I'll put a link in the in your module so you can read it yourself. But the basic idea, there's this guy who's worked with ventilators and respiratory things for a long time and working at Bellevue Hospital in New York and talking to the people who are on the front lines dealing with this. And what they're finding, that part of the problem with this COVID-19 COVID-19. So we already mentioned last class that this attacks your surfactant cells. And, surfactant is necessary to, normally in your alveoli, you've got that, surface tension of the water is trying to collapse the alveoli. And surfactant is like a detergent that can break that surface tension. It basically disrupts the attractions between the water molecules. So you have less of that surface tension and it's easier to inflate. The thing about COVID-19 that they are finding is one of the ways it's particularly dangerous is when you have this going on, it doesn't initially affect your lung compliance. That kind of stretchiness of your lungs. So even though things are going wrong and you aren't able to inflate your lungs properly because there's more resistance and the alveoli aren't blowing up all the way, when you exhale, the lungs still collapse and push out all the air that's in them. So you're not building up CO2. Right? So even though things are going wrong and actually you're not getting enough oxygen, you don't notice it because as we talked about in our lab last class, in general, your body doesn't monitor oxygen. It monitors CO2 to decide whether or not you feel like you need to breathe or not. So you aren't noticing, you don't notice anything's wrong. And you don't notice something is wrong for quite a while. I mean, there are other detectors in your body that can look at oxygen. I didn't mention, like in the main baroreceptors in your body that are adjusting your blood pressure are in your aorta and in your carotid arteries. Those places also have little chemoreceptors that are detecting carbon dioxide and oxygen. So your body can notice at some level there's not enough oxygen and maybe increase the depth and force of your breathing, but you're not noticing that something's wrong. You're not feeling out of breath. So what this guy in this article is saying was that people don't realize that they are having lung problems, but they're actually starting to push their lungs harder than they normally would. And plus their oxygen levels are going down and people actually start having all sorts of other issues because their overall oxygen levels are down. Like this is, I'm sure you've seen one of these. This thing right here, let me stop sharing just for a second here. This is a useful thing to know about, like a pulse oximeter. This little thing here basically shines light into your finger and can tell you what the percent of, or how much oxygen is in your blood. And we're going to talk about percent hemoglobin concentration in a few moments. Typically, if you're ventilating well, your blood should be somewhere around 98, 99% fully loaded up with oxygen. So if I do that here, and I just have it going, it's saying I am doing well. So I've actually literally been for the last week or so, rather than just taking my temperatures, look to see if there's a fever, if there's something wrong, making sure that the blood oxygen levels are also doing well so that there's no compromised lung function that you might not even notice is happening. So pulse oximeters, they're cheap and they're cool. Um, but so that is just this interesting thing. What they're finding is by the time the people get into the hospital because they actually notice something's wrong and that they're having trouble breathing, things are way further along than you would imagine, because actually the real problems and damage started happening much earlier when maybe they were feeling a little off or something, but they didn't actually realize that they were actually having pretty severely compromised lung issues way before they actually had any explicit sense something's going on. So by the time they get them on a ventilator, things are really gone south in their lungs already. So it's I'll link to the article, but I thought it was particularly interesting because it brings in a lot of the things we've been talking about, particularly surfactant, brings in the fact that carbon dioxide rather than oxygen is what makes you feel like you need to breathe. It brings in the idea of lung compliance, that the way you exhale is by kind of relaxing, and that stretchiness kind of naturally makes the lungs smaller as the chest cavity relaxes. So, and I think it's like this disease gets less scary. In some ways it's more scary, but less scary if you kind of have more of an understanding of what it's actually doing. So
You know why one of the symptoms is losing your sense of taste or smell?
that I don't know.
Okay, I heard that one.
Yeah, no, I don't know. So let's finish up the respiratory. And I should also mention, I had actually one other thing I was going to say. Oh, where is it? Oh. Yeah, I can really put my, the official term for when you're not getting enough oxygen is hypoxia. And this is actually worth just bringing in because it brings in stuff we've talked about. One way you can have hypoxia, you can have anemic hypoxia, right? If you have some problem where your blood doesn't have the ability to carry the oxygen. We talked about you can have iron deficiency anemia or hemorrhagic anemia or pernicious anemia, anemia. So you can be hypoxic, your tissues aren't getting enough oxygen because your blood can't do the job to carry it. You can have ischemic hypoxia. Ischemia is when there's an actual lack of blood flow. So here you're just not getting enough blood flow to the tissues because there's some blockages or something else going on. And then there's what's called, hypoxemic. So this is where it's like, from bad ventilation, etc. This is when you just don't have enough oxygen loaded up on the blood that you got because, what we've just been talking about, for instance, in this idea of you're not properly ventilating because the surfactant isn't working and your normal breathing isn't inflating the lungs well. So, these are all different ways you can end up not having enough oxygen getting to your tissues. It could be the blood doesn't have the ability to deliver it well, or your blood isn't getting there, or you're not actually loading the blood with oxygen in the first place. So kind of just tying these things in together and, kind of showing also, I think it makes this class a little more fun in some ways when we can see like, oh, this is actually useful if you're going to go out there and actually want to work with people and try to make them better, is understanding how all these things relate to each other. So maybe the silver lining of this COVID thing is it makes people even more motivated and feel like, oh, okay, this is worth learning about. I don't know. Yeah, I'd rather not have it happening. Okay, so let's Now do the last part, this external respiration, sorry, internal respiration. So this was this idea that you're moving the oxygen that's coming into the tissues. So the partial pressure of oxygen is around 100 millimeters of mercury. As the blood enters into the tissues, it's around 40 millimeters of mercury. As it leaves the tissues, again, these are just all your cells. So what we're going to talk about is this process of delivering the oxygen to the tissue and how it's carried and what conditions allow for it to release and go here. So like 98.5% of O2 is carried by hemoglobin. There's a little itsy bit, like 1%, I think 1.5% kind of dissolved in your plasma, but almost all of it is carried by bound to hemoglobin. Obviously, a lot of the oxygen's being released here. What we're going to look is some of the particular qualities of how oxygen and hemoglobin bind together and what kind of conditions affect that binding and affect the affinity of hemoglobin for oxygen, how tightly it holds on or lets go. So we're going to look more closely at this particular little relationship here, oxygen bound to hemoglobin. And we're going to do the famous oxygen hemoglobin dissociation curve. So let's do that. Maybe I'll use my typing tool to So this is basically going to talk about what kind of conditions does hemoglobin and oxygen stay connected, and what conditions do they separate and the oxygen is released to enter into the tissue. And we will do this as a graph. Oh, let's do black. So our, I'm going to go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this axis here, this is going to be percent saturation with oxygen. Let me make it smaller so it fits on here. So basically this is saying how much of the total load possible. We talked about how a hemoglobin molecule combined 4 oxygen molecules, and every red blood cell has 250 million hemoglobins. And, you know, so if every hemoglobin is attached to its full complement of as much oxygen as possible, we would say that the blood is currently at 100% saturation. It just means that the blood is carrying as much as possible given the, the nature of how oxygen binds hemoglobin. 0 would mean there is no oxygen attached. 100% means it's topped off completely, like the bucket is filled to the brim. You can't add any more oxygen to carry. All right, so then on our other axis, this is going to be partial pressure of oxygen. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Obviously, this is in millimeters of mercury. So So 40 millimeters of mercury, that is the typical conditions when the blood is leaving the capillary bed. So again, if we go back to that drawing I just drew here, as the blood is leaving the capillary bed and delivered its oxygen, it's down to about 40 millimeters of mercury oxygen. And so we want to see how much oxygen is actually still attached to the hemoglobin under this condition. And it's not what you think, probably. I'm going to do my best to draw this. So obviously here, let's look here at 100. If I have the surrounding partial pressure at 100, let's go back to this. If it was 100 millimeters of mercury out here, and the blood that's coming in is already at 100 millimeters of mercury, is the oxygen going to let go of the blood at all? There's no pressure difference, right? It's not. So if we are looking at 100 millimeters of mercury, all the oxygen stays attached to the blood. If we have 0 millimeters of mercury, and we think about, it's like if there was 0 out here, pretty much all the oxygen is going to leave and go into the surrounding tissue, right? If you have 0, there's this huge pressure gradient, partial pressure of oxygen for oxygen to leave the blood and move away. So if we look at our graph here, it makes sense that at 0 partial pressure of oxygen outside, that the blood has let go of all the oxygen. There's no oxygen left attached to the hemoglobin. If it's 100 millimeters of mercury around, then the oxygen is still stuck on the hemoglobin. It hasn't let go at all. What is non-intuitive here is this. So I'm drawing this line at 75% here. Do my best. The curve looks like this. And let's think about what this means. This means at 40 millimeters of mercury, which is what the blood is at as it's leaving the tissue and heading back to the heart, how much oxygen is still bound to it?
75%.
75%. It's kind of, so basically when we're coming back to this picture here, our little, I don't know what his name is, little Benny the blood drop is here, coming in, delivering his load of oxygen to the tissues. And then, we draw him as, oh, now he's blue and he's going to go pick up more oxygen. the reality is little Benny the blood drop, he still has 75% of his oxygen attached as he's going back to the heart to go to the pulmonary circuit. So when the blood returns into the vena cava, it's still typically bound to 75% of the original oxygen that it had when it started. It would be like if you filled up a bucket and you went to the fire and you just spilled out a quarter of it and then went back and topped the bucket back off and then went back, you spilled out just 25% and then went back and topped it all off again. So does that make sense? Again, it's a little non-intuitive. What is the use of that? Why would we have, so typically when the blood is returning to the heart after delivering oxygen, it's still bound to like 75% of its original load of oxygen. What possible use could this be? Someone?
The oxygen for the heart?
No, the heart is getting its supplied directly off the coronary arteries. So it's not waiting for blood to come back or anything from the circulation, right? It's getting a direct arterial feed off the base of the aorta for the coronary arteries.
Is it a protection mechanism so the brain gets enough?
No, again, the brain does, we're going to talk about that a little more. The brain will always get as much blood as it needs if possible. But again, this is blood returning to the heart after it's delivered oxygen. This isn't about initially sending blood out to the tissues. Okay, so basically you've got this great reserve. Like what if the conditions are in your tissues, there's really high metabolic demands at the moment, and you actually need way more oxygen than you normally would just at rest. It turns out the blood has lots of extra oxygen in reserve that it can actually let go of if it needs to. So for instance, what kind of conditions are going to, do you think, so let's talk about what, I'm going to use my type tool again. So what kind of conditions do you think would be in play that would cause the hemoglobin to have less affinity? If it has less affinity, I didn't mean to do that. Less affinity means you're going to let go of more oxygen. More oxygen is going to go into the tissue. So if, for instance, under these same conditions, maybe only this much is left. We would call this, maybe I'll use a different color here. You can move the whole curve to the right. Under any particular condition, less oxygen is left on the blood as it's returning to the heart. We've released more into the tissues. What kind of conditions do you think would facilitate or would necessitate that? What kind of things go on if you have increased metabolic activity in your tissues?
More CO2.
Totally. Increase of CO2. And for reasons we're going to see in a few moments, that is also going to mean increased pH. because carbon dioxide actually gets converted into carbonic acid. It has an official name, like the Bohr effect. Increased pH is going to cause more oxygen to be released from the hemoglobin into the tissue. What other things do you think? How is the condition going to, how's the condition in the tissue going to change if there's a lot of metabolic activity? More CO2, definitely. Think about yourself. If you're just being really active, what does it feel like to be you?
You need more energy.
So as you're burning more energy, what kind of, how do things change? How do you notice it? It's like,
Your fatigue.
no,
Temperature increase?
yeah,
Body
that's
gets
what I'm
warmer?
looking for. Temperature increase. Increased temperature. I'm obviously not being so great at fishing for this here. So increased temperature in the tissues. That is another sign that there's increased metabolic activity. That's going to cause the hemoglobin to let go of oxygen more easily, decrease affinity, and therefore allow more oxygen to enter into the tissues. There's one other thing which is called BPG. Its actual name is 2,3-biphosphoglycerate. Don't worry about its actual name, but this is basically made by red blood cells as they go through glycolysis. It's actually one of the intermediates. It's basically a indicator that the cells are doing more metabolism or higher rates of metabolism. And it ends up going up if you have like increased growth hormone or increased thyroid hormone or the catecholamines like the adrenaline and stuff. All those things increase metabolic activity, which increases the amount of this molecule. So that's another thing that tells the hemoglobin to let go of more. So this is basically goes up in response to increased metabolism. So does this make sense? Are people clear about this? Again, the first time I saw this, I was like, what? You know, the fact that the blood is returning to the heart when you're just in normal conditions with still 75% of the blood, 75% of the oxygen still attached. But it means that if there is increased metabolic activity in the tissues, you can actually let go of a lot more oxygen to deliver, big increases of oxygen to the tissues. And again, increased carbon dioxide, not increased pH, holy crap, decreased pH. More acidity, oh my God. So increased carbon dioxide or decreased pH, increase in temperature, increase in this BPG, which is a sign of increased metabolic activity, all decreased affinity of hemoglobin for oxygen, meaning more oxygen enters the tissues. So that's oxygen. We've talked about kind of the delivery of oxygen. We should also talk about how carbon dioxide is transported in the blood. So there's actually going to be three main ways. And one of them I mentioned when we first talked about hemoglobin, right? Hemoglobin binds oxygen. We've just been talking about it. but it can also bind carbon dioxide. So maybe like 30% is bound to hemoglobin. Hemoglobin isn't just for oxygen. The carbon dioxide binds a different site. It's on that protein globin subunit. It's not bound onto the heme group like the oxygen, but it's still bound to the hemoglobin molecule. You know, and again, the binding of carbon dioxide to hemoglobin is one of the things that affects hemoglobin's affinity for oxygen. And we just talked about how CO2 levels affect how tightly the hemoglobin is letting go of, or holding on or letting go of oxygen. So here's this kind of this direct connection here. CO2 binding onto the hemoglobin actually has effects on the molecule that make it hold on to oxygen less tightly. Maybe 10%. You know, I'm doing like somewhere dissolved in plasma. You know, carbon dioxide dissolves in water way better than, or way easier than oxygen. So it's basically soda water, right? Soda water is just carbon dioxide dissolved in water. You've all experienced fizzy water, Coca-Cola or Perrier or whatever. So you've got some of the CO2 is just dissolved in your plasma just as a gas in water. Obviously that leaves the lion's share, you know, is as bicarbonate ion. So let's talk about that. So basically CO2 plus water becomes carbonic acid. And there is an enzyme in your blood, but if your RBCs that is making this happen. The enzyme is called carbonic anhydrase. What the? Oh, hold on, it just auto-corrected it into Bizarro land. There we go. So carbonic anhydrase is this enzyme that converts CO2 in water into H2CO3 carbonic acid. And again, it's in equilibrium. It's going to depend on the concentrations of the reactants and products, which direction this goes. It goes both ways easily. We've seen what happens to carbonic acid. It's a weak acid. We talked about it when we initially talked about buffers in the first week of class. This just dissociates into H plus HCO3 minus. This is what we call the bicarbonate ion. So when you have carbon dioxide in the blood, some of it's bound to the hemoglobin, some just dissolves in the plasma, dissolves in the water. But the majority of it gets converted into carbonic acid, which then dissociates into this. So we have this bicarbonate ions floating around. One thing to notice here is the more CO2 you've got, the more acidic things become. We're gonna see when we get into pH balance a little later. A big part of your pH balance is by adjusting your pulmonary ventilation. If you are breathing really deep and hyperventilating, blowing off lots of CO2, moving the reaction this way, you're going to become more alkaline. If you breathe more shallowly, let the CO2 build up in your body, then the reaction is going to be more on this side and you're going to actually be more acidic. So actually one of the medium range controls for adjusting pH is just by changing kind of depth and rate of breathing. We'll talk a little more about that. But you can affect your pH like up or down by 0.2 just by doubling or halving your pulmonary ventilation. So this pH quality here actually is important. Obviously, if there's a lot of CO2, like when you're in your tissues, the reaction goes this way and you end up getting bicarbonate in your bloodstream. When you get back into the lungs, where you have a low partial pressure of CO2 compared, you actually have the reaction go this way and the bicarbonate reconfigures back into this side and then the CO2 can get released. So this is all, it goes back or forth. It goes, this way if you have a high concentration of CO2 around compared to the blood, so you get more bicarbonate coming in, but it'll go this way on the opposite when you get back to the lungs and want to release the CO2. So transporting CO2 in the blood, more complicated in some ways than oxygen. Oxygen is just, what's the answer? How is oxygen transported? Bound to hemoglobin. If there's a question on your exam, how is CO2 transported in the blood? You've got like 3 different things going on here. There's probably going to be a question about this on your exam. Exam is on Monday, and we'll go through the cardiovascular system and the respiratory system.
Do we know this formula?
What's that?
Should we know this whole formula down here?
Yeah, totally. Yeah, this is something that should look familiar already from when we talked about buffering. This is also the main buffer system in your blood, right? I mean, I can draw it again, maybe even, again, water plus carbon dioxide reversibly becomes carbonic acid, reversibly dissociates into H plus and bicarbonate ion. You should know this because it plays a few very important roles. It's how we transport carbon dioxide in the blood. It's going to be important for understanding pH regulation. It's also important for understanding the primary buffering system in your plasma. So, and it's pretty straightforward. There's not much to it. Again, the enzyme here is carbonic anhydrase. That actually makes that conversion from water and CO2 into carbonic acid. So he's like, oh, I see. Looks good.
Could you possibly explain it one more time, real quick or briefly?
Sure.
Yeah, I'm a little confused still.
So carbon dioxide enters into your blood. And then it turns out there is this reaction that is catalyzed by this enzyme. This is my enzyme, which is the red blood cells have got. So it's all over the place here. This enzyme takes a H2O and a CO2 and assembles them into this thing, which we call carbonic acid. Right, so we have the 2 ingredients are just waiting. There's lots of water, just 92% of your plasma, and the CO2 is coming in because of the pressure gradient. Then once they are together in the presence of this enzyme, it creates carbonic acid. This is a weak acid. But as an acid, it in water is going to dissociate. It's going to naturally break apart, just like if you put salt in water, because of Na plus and a Cl minus. When you put this H2CO3 into water, it breaks apart into an H plus into a, this is a bicarbonate ion. So now these two things are floating in water. This is kind of like our intermediate. And actually, and not all of it actually is dissociated, but so now we're here. And so as long as we have like a really large amount of the CO2, it's going to drive the reaction this way, right? In any kind of reaction, if it's going to, the direction that things go and where an equilibrium settles is going to depend on the concentrations of the different reactants and products. So if we have a lot of CO2, we're going to be moving it, having the reaction go this way and making more of this bicarbonate. If we have low CO2, like when you're coming back to the lungs to get rid of the carbon dioxide that's carried in your blood, right? At that point, and I talked briefly, like partial pressure of CO2 in your venous blood is around like 45 millimeters of mercury, but inside the alveoli, it's only 40. So the carbon dioxide is leaving. And as the carbon dioxide leaves, that's going to make this reaction keep going this direction. So more of this is going to just regroup, become this, and then come apart, and the CO2 will outgas.
And then you said something about how when it's the pH is low, then more oxygen will be released into the tissue.
Yeah, so that goes back to this. Remember we said increased CO2 means, right, normally, let's say at 40 millimeters of mercury, the blood only releases 25% of its bound hemoglobin.
Mm-hmm.
But in this situation where there's a lot more CO2 around, maybe now we're releasing 50% of the bound hemoglobin. So the amount of CO2 around is one of the important things that affects how tightly the hemoglobin holds on to the oxygen under any particular condition.
Does this pH have to do with that equation?
Yeah, absolutely.
So where would the low, so if there's a high concentration of CO2, then that means there's a low pH?
Yes.
Okay.
Yeah, CO2, in fact, the respiratory, I'm going to talk really briefly now about the respiratory centers in the brain to just finish up things.
For the first part of that equation, though, I thought you said it was basic, which means there's a high pH.
Right, but then remember I said, oh, and yeah, no, if you take a look at it now.
Oh, is it switched?
See that?
Oh, so it would be more acidic.
Yeah. No. So you were paying attention and I totally spazzed. The pH goes down when CO2 goes up.
Okay.
Yeah,
So
I'm going to
the
have
part of that equation should be acidic. The CO2 plus H2O, that's more acidic.
yeah, I mean, let's go back to it. Yeah, we're having more H pluses, right? That's the definition of acidic, increasing the concentration of H pluses. By definition, we're becoming acidic by adding in H pluses.
Okay.
So an acid is something that is going to be contributing protons. So, all right, so how is this making sense now?
Yeah.
Yes.
Cool. All right, so now let's go back. So respiratory control. I am not going to get into the nitty-gritty details of this. I'm going to say in the pons and the medulla, mainly driven by CO2 levels. Part of that works is actually the CSF does not have the same buffering that your blood has. So that kind of acidity of the carbon dioxide is actually going to be changing pHs, and the pH is going to be telling the medulla, oh, we need to ventilate more because carbon dioxide is building up. But basically monitoring CO2 levels, like I said, you can actually have, in fact, this happened in lab when I was in grad school. Somebody tested it out, just breathing pure nitrogen. Ventilating, you never feel anything's wrong, and all of a sudden you heard just like this clank as he head planted on the tank because he passed out and didn't know anything was going wrong until because he didn't realize the oxygen levels were going down because there were no alarms going off, because as long as you're ventilating, CO2 is being blown off and CO2 is not building up, and you don't realize that you're actually not getting enough oxygen. So that is important too. That's a super important thing. And there are supposedly people who died in the astronaut program walking into a room that had been flooded with nitrogen and had no idea anything's wrong until like they passed out and suffocated. Actually, there's a bunch of stories about, I can tell you a bunch of stories about people who died because they don't know they're suffocating. But in the interest of time, let's keep moving. And actually, there's lots of cool things. If you can hit me up later, we can talk about diving in shallow water blackouts and things like that. Normally, if I would talk a little more about that because that's also very much related to this. But again, everything's going so much slower now. Let's talk about just a couple of pathologies. There's things like emphysema. Emphysema, you start getting the walls of the alveoli breaking down. Good, I'm gonna type because in my trying to go fast, I'm making it worse. All right, so you can think about why is that bad? So we have our little alveolar sacs and the kind of stretchiness. Now, all of a sudden, we are kind of losing the walls. We have less wall than we used to. Why is that going to be bad?
Less gas exchange.
Yeah, way less. You have less surface area for gas exchange. Remember, surface area, when we talked about diffusion rate, things that affect diffusion rate, one is just the surface area across which diffusion can happen. If you have less, less area, surface area for gas exchange, you're going to have to be less efficient at moving oxygen into your blood. Having the lung tissue lose its elasticity, that means the compliance is gone down. That means when you just relax your muscles in your chest and the chest wall gets smaller, normally the lungs naturally want to get smaller, and they're going to get smaller too because that's what they've been trying to do anyway. They've just been held open by that suction with the pleura. But when you have emphysema, the lungs don't naturally collapse down the way they normally would. So often people with emphysema have to actually be more active in their exhalation. So they have to work to exhale as well as to inhale. And also because of that, because the exhalation is not as natural and efficient, they tend to have increased levels of carbon dioxide in their body all the time because they're not ventilating well, they're not blowing out the carbon dioxide. One of the things that's kind of fascinating in emphysema is, you know, it's one times you can have a bit of an exception to what I just told you. I was saying that basically you look at CO2 levels to drive your breathing. People who have like emphysema where chronically elevated CO2 levels are happening, they actually do have their body like shift a little bit and start using like the oxygen sensors in their, you know, in their aorta and their carotid arteries to actually start driving some of their breathing and tell them they need to breathe or not. So you can actually get like, you know, I don't know how true it is. One of the tales is like if you have somebody with emphysema and you put an oxygen mask on them, sometimes they'll stop breathing because their bodies have, normally if you put an oxygen mask on somebody, it doesn't affect their ventilation because they still feel like they need to breathe as the CO2 is building up in their lungs. So breathing pure oxygen is not going to make you stop breathing. But if you have emphysema and you give them an oxygen mask, because their system has recalibrated to actually measure, like, do I have enough oxygen? If you give them pure oxygen, supposedly they'll like stop breathing or they can stop breathing. So that's kind of fascinating to think about. What other things do I have here in terms of pathologies? Yeah, asthma, we already talked about. Asthma is just kind of this narrowing of the bronchial passages. Again, reduced diameter means increased resistance. Increased resistance then means it's harder to get your flow going. You need more pressure to get flow going. What other things do I have here? Pneumonia. Pneumonia tends to be when you have, actually, I think it spelled it wrong. It doesn't matter. You know, as you get fluid buildup in your lungs, and that's going to make it harder for gas exchange because now the gas has a larger distance to have to try to diffuse, less surface area. It can make it harder to start inflating things. So pneumonia, and one of the fascinating things about pneumonia, this was another thing like I found interesting as I was learning about this stuff. A lot of times people die, like when they end up in the hospital and old and sick, and ultimately it's like they died of pneumonia. But typically, you know, this is, you know, and it's caused by lots of different things. But typically, like when you find some old person's died of pneumonia, it's actually like lots of things have been shutting down and their body's having an inability to fight things in general, which is different. I mean, now this COVID thing actually is, I think, genuinely taking people who are okay and killing them. But outside of the COVID world, this often is kind of something that happens kind of secondary as your body has trouble, like, you know, fighting things in general. A lot of times, if you have respiratory infections, you can fight it and get over it. What else? Rhinitis. I love the name rhinitis. What is rhinitis? right? Rhino means nose. So it's rhinitis. Rhinitis is like what you got when you got like a common cold. It's just inflammation of the upper respiratory passages. The official name for the virus that gives you a cold is actually rhinovirus. Coronavirus is the one that's getting all the press. Rhinovirus has a cool name, and it's the one that gives you the common cold. Bronchitis is another. Bronchitis would be lower respiratory inflammation. So if you have trouble, lower respiratory tract would be bronchitis. Rhinitis is upper respiratory tract, and sneezing and sniffling and all that. So, have here, become inflamed, excessive mucus, etc. So let me see, I'm looking here. Any questions about any of this?
Just curious, in pathologies, how would TB work?
So I'd have to look at that. I mean, that's a bacterium,
Okay.
and I'm not sure exactly. I think it causes scarring and stuff in the lung tissue. I can look that up when we take a break and figure that out. It's definitely, it's a bacteria that, and obviously it causes scarring because you can tell by doing x-rays to see if somebody's had it. But I don't know much more than that. So it's 9:30. I do want to introduce the urinary system before we're done today. So I think what we're going to probably do is take a break. I'll probably introduce the urinary system, and we'll do a little break, and then we'll do the lab, which, again, particularly because we're not actually going in and having to hook all sorts of stuff up, it's going to go way faster than it normally would. But the lab will be fun. The lab used to be fun. You actually get to breathe into things and do fun stuff, but you'll get to see Becky breathing into something. And let me just remind you, I'm going to write it here just so nobody forgets. Exam 3 on Monday. Make sure 4/27. Covering cardiovascular and respiratory. So everything up to what we just mentioned right now is fair game. This will be using that Proctorio. Now make sure, I'll remind you all, but you have to make sure you open up your canvas in Chrome with the Proctorio thing installed. And I've found most of the problems where people have issues are when they actually don't open up the program in Chrome. Then it starts asking for a password, and normally Proctorio will supply that. The good thing about using Proctorio, I know one of the issues, again, people had was seeing questions come one at a time is unpleasant and disconcerting, and you can't go back. With the Proctorio, I have the setting where you can roll back. So you'll all be taking the exam at the same time, but you can also, you don't have to like see one question and then it goes away forever. You can actually double check you're happy with the whole exam before you submit it. So hopefully that makes people feel a little less stressed about things.
Dr. E?
Uh-huh.
Are you thinking 8 a.m. start time for the exam or lab time?
No, 8 a.m. I think makes sense,
Okay, great, thanks.
or 8:10. Make sure we get through things. I'm gonna spend a little bit of time right now introducing the urinary system. We're not gonna go through the whole thing, but maybe spend a half an hour just kind of laying out some of the basic concepts. So we just kind of keep that rolling, and then the lab is not gonna be nearly as long as it normally would take, again, because a lot of what we do in the lab is actually physically setting things up and all that. So I'm still recording. So what we're gonna do is introduce the urinary system. And
This system won't be on our test though, will it?
no, this will not be on your test. This is for your exam 4, which will be during finals week. So like I said, I'm digging into this now so we kind of get it rolling so we don't run out of time by the end. But not on exam 3. Like, wow, so glad it's not on exam 3. So let's instead let us talk about what it does. So what are functions of the urinary system? We've already seen it in play. What did we see it playing a critical role in the cardiovascular system already?
Flushes out waste products.
It does do that, although let's go back to the cardiovascular system and then we'll come back to that one. What was it doing that was part of our discussion of blood pressure and all that was so critical?
Regulating blood volume.
Yeah, exactly. So big part is water balance. And water balance is also basically blood volume. So that's a big part of the urinary system, is holding on to or letting go of water based on your body's needs and how much you've been drinking and what is your set point should be. So water balance is going to be a huge part. And again, we saw that in, remember, the renin-angiotensin mechanism, which will definitely be on your exam. Absolutely. You should understand renin-angiotensin mechanism and how it relates to the kidneys. How did, in fact, just as a brief, quick review, how did the renin-angiotensin mechanism involve the kidneys?
Well, the kidney is what secretes the renin, right?
So exactly, the renin starts by being secreted by the kidneys. And then as things go on, how do the kidneys get activated?
Blood pressure drops.
Actually, it's the opposite. It's supposed to bring the blood pressure up. It's activated when your blood pressure drops. People have been putting off their studying, it seems. Renin-angiotensin mechanism. Anybody? Remember, renin then catalyzes angiotensinogen into angiotensin 1, which then gets converted into angiotensin 2 by ACE, that angiotensin converting enzyme, which is localized in the lung tissue. And then angiotensin 2 is a vasoconstrictor, but then also activates the release of ADH by the pituitary and aldosterone by the adrenal cortex. And both aldosterone and ADH work at the kidneys. Aldosterone to increase salt retention and ADH to increase water retention. And together they maximize keeping water in the body to keep blood volume up, to keep blood pressure up. That should all be like really, really familiar before Monday, 8 a.m. I'll say. It's one of the, I brought it up as one of the key mechanisms in regulating blood pressure. You should definitely understand the big picture and then the smaller pictures of why each of those parts are important for affecting blood pressure. And in fact, ACE, remember ACE? What was ACE?
Does it stop?
Angiotensin converting enzyme.
Right, it's that angiotensin converting enzyme. That was the target out of a whole lot of blood pressure meds, is to inhibit ACE and therefore kind of stop this whole thing. And in fact, ACE comes up, ACE is the ACE we've been talking about is the ACE1, but there's another one of the angiotensin converting enzymes anchored on the lung epithelial cells is actually the portal that the coronavirus uses to get into your cells to cause COVID-19. It actually uses an enzyme bound on the epithelial cells in your lungs. So this thing, again, there's ACE1, it's ACE2 are the ones for the COVID, but it's actually the portal that the virus can anchor onto to enter the cells. But that's not part of the urinary system. But you should, this is super important. You should definitely know renin-angiotensin mechanism. So water balance, somebody said, getting rid of wastes. There's all sorts of waste. There's metabolic byproducts from all the different things going on in your body. There's urea. We talked about urea getting formed during like protein breakdown as you deaminate the amino acids and stuff. There's, whatever you have that you need to get rid of, this is a way to actually get it out of your body, kind of excrete them. What are other things the urinary system does? Nobody? So aldosterone, what did aldosterone do? We talked about aldosterone in the renin-angiotensin mechanism. What is it adjusting?
Salt.
So this is a big part of your electrolyte balance as well. Exactly. So we've seen since the beginning that the levels of sodium and potassium and chlorine and all that are critical for your cells functioning. All of those voltages across the membranes, all of the excitation of neurons and muscles, all depends on having the appropriate concentrations of ions inside and outside the cells. So it's critical that you have the correct salt balance in your body. And so the urinary system is going to be doing that, either holding it in or peeing out more if you've got too much. So they're going to be critical in electrolyte balance. Another one I'll throw in here, pH balance. You know, in fact, balal, I just made up a new word. We've seen buffers can affect pH, but they're kind of temporary based upon how much of the weak acids and bases you have to associate or reassociate. We saw, I just mentioned, respiratory system, changing your ventilation can change your pH. If you hyper or hypoventilate, your pH will go up or down. But that's also something that is not that's something you're adjusting from resting. The only main long-term control to set your resting pH balance is actually your urinary system. Either hold on to or pee out the excess hydrogen ions and bicarbonate ions and stuff like that. So urinary system is going to be the primary long-term control of your pH. So we'll see it there. So it's doing lots of things. Yes, it does get rid of waste, but that's just one part of many super important things it does. Like when I think of it, it's like water balance, it's electrolyte balance, it's pH balance. So that is the urinary system. This is a slide from your book. What I'll do is I realize I've been using some of these PowerPoints. I don't think I have them up. I'll upload any PowerPoint that I'm using in the lecture. I'll upload onto the website, onto the module, so you have access to all of these things. So when we think about the urinary system, the kidneys, you've got two. Luckily, they're both doing the same thing. And either one alone, actually, assuming they're healthy kidneys, has the ability to do all the stuff that we've been talking about. So you don't need both kidneys to stay healthy and do all your water and electrolyte balance and get rid of wastes, which means that you can donate a kidney to somebody and you'll be okay, assuming that your remaining kidney stays healthy. We're gonna spend a lot of time looking at the details in the kidney that are doing all that stuff we're talking about. They're being fed by the renal arteries. One of the things that is fascinating here is 20 to 25% of your cardiac output is entering into the kidneys. Into those renal arteries. So how much blood is that? How many liters per minute are going into your kidneys?
About one.
Yeah, exactly. Normal cardiac output's about 5 liters per minute. So we have 1 to 1.25 liters per minute of blood entering into your kidneys. That's a huge amount of blood. Obviously, your kidney tissue doesn't need that much blood just for its oxygen and for its nutrients. So most of that is so the blood can be processed, and that's where we're going to be doing all that stuff, doing all the balancing, getting rid of wastes and things. So we'll be spending, again, a lot of time looking at what's going on in the kidneys. And again, we'll come back to this number in a little bit when we go into the details of how the kidneys work. But you've got about 1 to 1.25 liters per minute entering into the kidneys. So it's a huge, again, up to 1/5 of your overall blood flow is just coursing through the kidneys. The ureter is what carries away whatever is going to be your waste product. Excess water, excess salts that you don't need, wastes, things like urea, things that you don't want in your body. So the things that finally come out here as urine, that's going to be all the stuff that you don't want anymore. Again, it could be extra salt, extra water, and waste products. We have the bladder, which we'll talk about in a few moments. A nice little added feature, so you actually can store the urine until there's an appropriate time and place to actually like relieve yourself and get rid of it. You know, as opposed to like, you know, birds basically just like kind of let it go whenever, so they don't start building up a lot of weight and make it hard to fly. So we'll talk about that. Urethra, whack. We'll talk about the urethra. The urethra will come up. In the female, the urethra is just kind of short. It's like 1.5 inches or so. It just ends up right in the vulva. In the guy, the urethra will be more involved because it's going to also be kind of the main place where you're mixing up all of the sperm and all the different secretions that make up the semen. So when we get to the reproductive system, the male urethra will actually be doing a lot of other stuff versus just conveying urine out of the body. But in the woman, the urethra is just a little tube from the bladder to outside of the world. All right. So we're going to focus just with this kind of big picture. Kidneys and bladder and ureters and urethra, and as well as a little bit of the control, the little sphincters that let you pee or not pee. And then we'll introduce the basic processes going on in the kidney. The kidneys are made out of like 1 million of these things called nephrons. These nephrons are these big, crazy tubey things. There are all these, there's proximal convoluted tubules and distal convoluted tubules and loops of Henle and collecting ducts and glomerular. There's all these parts and pieces that we'll learn about. But for right now, you just got to know that there are these nephrons and that the nephrons are doing important work that I will introduce. So the nephrons are the kind of repeating subunits that make up the kidney that do all the cool stuff that we care about. Yeah, here you can see a real nephron is really kind of wacky. And we will get into this, but that will be next week. So let's get back to just more overview right now. Okay. Kidneys. It's worth mentioning that the kidneys are kind of enveloped in what's called the adipose capsule. Which actually helps kind of cushion them and hold them in place. You know, you have the ureters are carrying the urine out, you know, into your bladder. It's my urinary bladder. The reason why it's worth actually mentioning the capsule here is you can actually have a problem if somebody loses too much weight. Somebody is like anorexic or something, and you start having too much of this adipose disappear, you can have the whole kidney drop down. Drooping of anything is called ptosis. You can have ptosis of your eyelid as well if your eyelid droops weird. But so if the kidney droops, it can actually kink up the ureter here and make it harder for the urine to flow out. So ureters are the things that carry urine from the kidneys to the bladder. The urethra is the thing that carries the urine from the bladder to the outside world. Make sure you get those two words straight. Ureters, kidneys to the bladder, urethra, bladder to kind of the side of a tree or wherever you're peeing on. Urinary bladder has a lot of smooth muscle in its wall. Ultimately can fill up to about 1 liter or so. It can fill up the first couple of 100 milliliters. It doesn't even really stretch out. But as it keeps filling up, it starts stretching. There's little stretch receptors, and we need to add in our little sphincter muscles here. The reason you don't pee on yourself, at least typically, is you have an internal sphincter here. Which is made out of smooth muscle. If you remember, smooth muscle is the one that is involuntary. And then you have a second sphincter, which is going to be your external sphincter. Which is skeletal muscle. And it's basically the muscles of your pelvic floor. So this one you have control over, you can squeeze. Internal one you don't have control over. This is going to be, there's going to be a spinal reflex. Just like, remember we had spinal reflexes when we looked at hitting your knee, the stretch reflexes. This is another spinal reflex. You have stretch receptors in the wall here. Stretch receptors, those are going to detect the stimulus. The stimulus is going to be stretch of the bladder wall. As the bladder wall stretches, it talks to your, and this is a spinal reflex, it's in your spinal cord. Just like we had with the stretch reflex with your knee. And the response is going to be relax the internal sphincter. So this internal sphincter will relax, which then puts the pressure down to the next level. This is when you feel the urge to urinate, to pee. I gotta pee. I gotta go make wee-wee or whatever you're gonna say. So you're feeling, but because the external sphincter is skeletal muscle, is voluntary, you have a choice. So you can decide whether or not you want to relax that one and then let the stream flow out.
Can I ask a question?
For sure.
So you know how with elderly, sometimes it's harder for them to hold their urine in or they release it on accident more. Does the sphincter just become, is it like loose? You know what I mean? Is it thinner or something or?
Right. So, I mean,
what is
that's,
that?
yeah, so I definitely know that incontinence is really common in older people, and I'm not exactly sure what the mechanism is there. I can look into that, but I don't know.
Okay.
Exactly. But yeah,
Okay.
so I definitely know, like in small kids, until you are, toilet trained. And then as on the other side, when you get much older, people have trouble actually making this work properly. And I'm not sure why. What else we can talk about? Urinary bladder. The urethra, like I said, in the guy is going to be a lot longer, and it's going to be where you mix up all of the stuff for the semen, and it's going to go along the length of the penis. In a woman, it's only like 1.5 inches, and it ends up in the vulva right near everything else. Like, and it's short. It's much more likely in a woman that bacteria or something can manage to get up the urethra into the urinary tract and cause a UTI, a urinary tract infection. Partly because it's a just much shorter distance to go, partly because the urethral opening is actually not that far from the anus where you got all the fecal bacteria and stuff like that. So women are much more likely to get urinary tract infections than guys. UTI. And the other thing I'll just add in here, that in a clinical world, the words people use, like, you know, when you're just saying, I gotta pee, people talk about, oh, I gotta pee. Or even if you want to be a little more sophisticated, you know, there's urination. But if you are in a more clinical setting, what are the official words people use?
Mituration.
Yeah, micturition. So micturition, you should, that's a fancier, more clinical term for urination or peeing. The other word people often use is voiding. So these are terms you'll often see having to do with just emptying your bladder. Yeah, if you're just like kind of at some like concert and it's like, I'll be back in a second, I gotta go micturate. People are gonna like, what? Huh? But if you're in a clinical setting, they'll know what you're talking about. So that's all I want to say about this kind of bigger picture here. You know, and we're going to see a similar system to this internal and external sphincter when we get to the digestive system. In your digestive system, you also have an internal anal sphincter and an external anal sphincter. And as your rectum fills up with feces, you have a spinal reflex. As the rectum stretches out, filling up with feces, internal sphincter relaxes and you feel the urge to poop. And then again, luckily, you have external sphincters that give you a voluntary control over whether or not you actually go at the moment. So yay for voluntary sphincters. Now what I want to do is just overview. This is the last thing, and then we're going to stop after this. Overview of nephrons. And again, the nephron is what we call like the structural and functional unit of the kidney. So there's like a million of these just repeated again and again. So if you understand the basic activity of the nephron, you've got at least the core idea of how the kidney is accomplishing what it does. We'll look at the nephron in more detail, but for right now, I just want to introduce these ideas so they can percolate. And when I dig into the nephron proper, I feel like you've already seen the basic idea. This is going to involve another one of these portal systems where you go from a capillary bed into another capillary bed. If you remember hepatic portal system, you went into the capillaries of the digestive organs and then absorbed all of the nutrients and stuff and then delivered that into a second capillary bed, which was in the liver for processing, and then finally went back to the heart. This is going to be another one of these examples where you're going to go into one capillary bed, but then go into a second capillary bed before you come home to the heart. So we're going to start with what we call our afferent arteriole. So the afferent arteriole is just the thing that's feeding into this whole thing. It goes into this little ball of super leaky capillaries called the glomerulus. And then continues on and to become the afferent arteriole here. Efferent. Remember, efferent leaving. And this thing is called the glomerulus. Glomerulus means little ball. And this is a little ball of fenestrated capillaries. You know, what is a fenestrated capillary? That's something you should know for your test.
Leaky?
They're super leaky. So as the blood is coming in, you start having a whole bunch of water and dissolved solutes are leaking out from this glomerulus. So we call this first step here. This first step is called filtration. And the stuff that are small solutes include both wastes like urea, but also good stuff like glucose, as well as things like salts, which you might or might not want to keep in your body depending on your body's needs. So it's very indiscriminate. You're making all of this filtrate, and it includes good stuff and bad stuff. And in fact, the amount of filtrate that's happening, this is going to be 10% of the flow coming in. So, in fact, this is worth doing here. Here's the blood flow coming in and out. So let's think about this. What is the blood flow coming into, again, looking at all of the glomeruli and all the nephrons in both kidneys? We just mentioned that at the very beginning. What is the amount, what is the blood flow coming into the kidneys?
1 liter.
Yeah, 1 to 1. So let's say 1.25 liters per minute coming in. This is flow into the kidneys. So 10% of that means we are going to have 0.125 liters per minute of filtrate formed. Right, and if we do the math here, if we do like 0.125 liters per minute times 60 minutes in an hour times 24 hours in a day, this actually becomes 180 liters per day. So this filtrate that's getting formed in the nephrons, you're making 180 liters of it per day. So obviously this is not your urine. It's a huge volume. You're going to have to reabsorb 99% of that back into your body. Only like about 1.5, 2 liters of that's going to ultimately become the urine that comes out of your body. So you're making this filtrate, you're making a bunch of it, 180 liters per day. So we need to reabsorb 99% of the volume of the water, as well as reabsorb the nutrients and the salts and stuff that we want to keep. So we're going to capture this in what's called the renal tubule. And this efferent arteriole is now going to come back and become a second capillary bed right here. This is going to be my peritubular capillaries. So again, this is my second capillary bed now. And as the blood is, as the system's going, basically anything that you want to pull back into the body, you can transfer from the renal tubule back into the bloodstream here. Again, so this is called, this part here, this is going to be part 2. This is called tubular reabsorption. Basically, this is going to be, recover most water, nutrients, and whatever else you want to keep. In terms of salt, it depends on your body's needs. If you've just been eating a whole bunch of Doritos or something, you probably don't have to recover the salt and just pee it out. You got more than you need. Or else if you're low on salt, you're probably going to recover it, pull it back into your body. So exactly what is going to be reabsorbed depends on your body's needs to do the balancing, do the water balance, do the salt balance, right? The last
Can I ask
part,
that?
say it again.
I just had a question about dialysis, unless you're going to get into that later.
We'll do that later.
Okay.
Dialysis is going to be trying to take over the roles of the kidneys, but let's do that when we're done with the kidneys.
Alrighty.
All right, so the last part, there can still be stuff in the blood that you actually want to get rid of. So anything that you have that you really want to have exit the body, you can actually actively move out of the blood into the tubule. So anything that gets here is going to ultimately come out as pee down here, right?
Yeah. Now, did you
So
find a little,
this last piece here, this is going to be called tubular secretion. So basically, there could be whatever, it's excess urea, this or that, whatever you're trying to get rid of, your body can transfer it from the blood into the tubule, and anything that's in the tubule, by the time you get to the end of this whole process, is going to be excreted and you're going to pee out of your body. So this is the big picture that we're going to look at in more detail, but I kind of wanted you to just kind of see this whole thing. Are there any questions about the big picture here? Okay, and the urinary system is probably one of the kind of trickiest of all of the systems. Once we actually get into the real details of the nephron, it's going to make, you're going to see there's lots of things going on. I have to decide how complicated I want to get given our timing. But like we talked about drugs for blood pressure. Two of the drugs for blood pressure you should already understand. We talked about like the beta blockers. You know, beta blockers which slow down your heart, decrease contractility of your cardiac muscle, which both serve to reduce stroke volume, which are then going to decrease cardiac output and decrease blood pressure. We talked about ACE inhibitors, which stop the renin-angiotensin mechanism. That brings down your blood pressure for all those reasons, both inhibiting vasoconstriction and inhibiting increase in blood volume. Another kind of drug people often get for blood pressure are antidiuretics. For instance, the loop diuretics, the classics like Lasix and stuff like that, they all work right here. They work by messing with the ability of the nephron to reabsorb the water. So a larger percentage of this crazy amount of filtrate actually does leave your body and you pee out more, and that's going to help keep your blood pressure down. So by inhibiting the reabsorption using an like a loop diuretic like Lasix, you actually then will end up having more water leave your body because you're reabsorbing less of it before you get to the peeing stage. So that makes sense?
Is the purpose of the Lasix to just make all the nutrients and waste come out then?
It's more about the water. It's to increase the
Okay,
amount
so
of
releasing
water
the
loss.
water.
Yeah, it's to increase the water
Okay.
loss.
Okay.
So and by increasing water loss, you keep your blood volume down, which keeps your blood pressure down.
Perfect. Thank you.
Sure. All right, so it's 10:45. At 11, we are going to start the lab. So let's do